Welcome back to Biomindscape. Today we are going to know about the complete developmental pathway of frog embryo. It begins from the fertilization and end in the organ formation. First, what is happening here? Fertilized egg is present, the zygote. It have two poles, that is animal and vegetal pole. The frog egg, it is mesolithical, that is it has a moderate yolk which is inside this layer. Then yolk accumulate in the vegetal pole. It is present at the bottom. Then there is an animal pole which is have less yolk content. That is, it is poor in yolk and it divide faster. The dorsal ventral axis form after the sperm. It enter a point shifted to the cytoplasm. It form the further dorsal side, that is the gray crescent. Then earlier cell division takes place. What do we call it? Cleavage. So it is holoblastic but unequal. The animal pole, it is small, whereas the vegetal pole, it is larger. The animal pole, it is known as the smaller. It have small micromores, whereas vegetal pole, it have large micromores. Then this cleavage, it forms 2 cell, then 4 cell, then 8 cell, and then finally the morula like stage as shown from top to the left of the diagram. The reason is inequality. More yolk slows down the division in the vegetal pole. Then here a blastula stage takes place where it have animal pole cells like which are small, whereas vegetal pole it is large. And the animal pole it form the ectoderm, whereas the vegetal pole it form the endoderm. And it is labeled as the blastula in this diagram. It appears in hollow ball of many cell and it contains blastocele. This blastocele, it is a fluid filled cavity and it shifts toward the animal pole. Then the next step, what is happening here? It is the gastrulation. The most important transformation in earlier development is gastrulation. Here where the gastrulation begins, it is gastrula. And first involution of the marginal zone. Here cell at the equatorial it roll inwards and form the dorsal lip of blastopore, that is known as Spearman's organizer. Then blastopore formation takes place, a circular opening where a cell move inside the embryo, and archenteron formation takes place. It is a new cavity which is primitive gut formation and it replaces the blastocele. Then there is a yolk plug where large vegetal cells get exposed temporarily at the base pore, blastopore, and then there is a germ layer formation like ecto, meso and endoderm. So ectoderm it is present outside, mesoderm is in between, and endoderm it is inside and it is in the lining of the archenteron. Then there is a neural stage where it shows the brain plus spinal cord formation, and it is a stage where organogenesis begins. So it is a major step where the neural plate form from the dorsal ectoderm. The neural plate fold to form the neural tube which further give rise to the brain plus the spinal cord. Then mesoderm it differentiate into notochord and somoid which further form the muscle, whereas vertebrates. And endoderm becomes the gut tube. It shows that the neurula is the lower part of the diagram. Then organogenesis takes place which further give rise to the tadpole and then finally the frog. So here the neural development in the tadpole is eventually into an adult frog shown in the diagram, and the brain and spinal cord are clearly labeled and showing the origin of the nervous system from the ectoderm. So what does this diagram state? From frog embryo development, it starts from fertilization, then cleavage, formation, then blastula is formed, then gastrulation takes place, neurulation takes place, and organogenesis takes place, showing how ectoderm, mesoderm and endoderm they are established and they give rise to the brain, spinal cord, gut and other organs. So for knowing such concepts, follow Biomindscape.